Unlock the future of pathology with Pathology News, your ultimate source for cutting-edge insights, expert analysis, and the latest breakthroughs in digital and computational pathology. Don't just stay informed, stay ahead.
Hello and welcome to Pathology News. Today we're coming to you from the city of Cambridge here in the UK, and we're actually at the Cambridge Institute of Cancer Research. Now, today's conversation is going to be focused on digital pathology and spatial biology and how the Cancer Research UK team, or more specifically the Cambridge Institute of Cancer Research UK team, are using those techniques in their research and practices here. Now, as well as that, we're also going to be covering a few current topics, including scanner performances and colour standardisation. So to explore all of this in some more detail, I'm joined by Dr. Julia Jones, who's the Scientific Manager for In Situ Hybridisation here, Elsa Santos, who's the Senior Scientific Associate, and Cara Brodie, who's also the Senior Scientific Associate. Now, as new guests on our podcast, I was hoping we could start by you telling us a little bit about yourselves. Julia, can we start with you?
Yes, hi, thanks very much for inviting us to your podcast. So I actually sort of came to the CRUK Cambridge Institute back in 2007 when the building opened, fresh from my PhD, which had been in neuroscience, where I'd sort of specialised or really sort of had a focus on in situ hybridisation. So when they were looking at kind of opening an in situ hybridisation service here, I thought, oh, that sounds like it was written for me.
Yeah.
And I've been here ever since. So that's kind of 19 years building up the in situ hybridisation service and always kind of following the recent advances, trying to keep up with what's going on in the in situ hybridisation world.
I suppose, digital pathology really only sits in the last 15 years. So you've seen the kind of advent of it, and you're still here now. So you've seen it from start to, well, not finish, but wherever we are now as well. Yeah,
exactly. And I think we've always been like maybe slightly ahead of the times in that we started out with scanners right from the start back in sort of 2007.
Elsa, what about you?
Well, for me, it's been a long journey. So I started my histology journey in around 15 years ago back in Portugal. So basically, after I finished my biology degree, I just wasn't sure what route to take. And I just signed up for this degree that's kind of specialized in histology. So once I finished, so it's quite difficult to find jobs in Portugal currently because there's very scarce jobs for the amount of people that get the degrees. So I just decided at the time I'll move to the UK, where there's a lot of demand for histology technicians. And then I was 6 months working in a routine histology lab, but I was always passionate about science, so I thought I would like to be more involved with research setting, and this job came up at the CRUK, so I just applied for it as a research assistant. And and then I was being line managed by Cara and mentored by Cara, which was covering the scanning services at the time. So I learned loads from Cara, and it became too much for Cara to then take on everything. So basically, I was invited to help and cover the scanning services, and that's how I became the senior from that area.
Thank you. And Cara, then, what about you?
Yeah, so I did my degree and then moved back to Cambridge because I was like, well, this is where I'm going to get some lab experience. So I was lucky that we lived around here. Got a job as a lab technician for the University of Cambridge, then got made redundant because the funding ran out. And then, applied for the research assistant job here. I was lined by Julia. Then the role of the senior in the scanning and image analysis came up. So I was like, it's mine. Applied for it and got it. So it started off with just managing 2 scanners, then it grew and grew and grew, and then...
No, I'm glad you mentioned image analysis, because obviously that's going to be a big component of today's conversation. To start with then, I was hoping we could learn a little bit more about what happens here at CI UK. You know, how are you guys using digital pathology and spatial biology?
Well, if I tell you like a little bit about the CI UK Cambridge Institute.
Yeah.
So we are a department of the University of Cambridge, and there are 17 research groups here.
Yeah.
And 12 core facilities. They're all focusing on different areas of cancer research.
Yeah.
And the core facilities all specialising in particular kind of, you know, their expertise. Sort of proteomics, microscopy, us histopathology. There's also sort of bioinformatics and many more.
Yeah.
And so our kind of core facility is really supporting those different research groups with all of their histology needs, helping them to...
So there's a lot of collaboration between
Exactly.
centres, I guess.
Yeah, that's right.
So yeah, so each of the cores sort of host the sort of high-end pieces of equipment. So obviously we are funded by charity money.
Yeah.
So as a result, if we centralise these high-end pieces of equipment, they're maintained to a high standard. They last a lot longer, and also everybody within the institute has access to them rather than being associated to a particular group. They get then shared across the institute.
Yeah.
That's
Yeah.
right.
Okay.
And then run by experts who can advise those, you know, those researchers on the best way to set up their experiment and to run it, and you know, for controls and so on.
Obviously, we're quite lucky to be at the front line of all of this and have like the support and the funding to trial new technologies. So I say we, but it's been mostly Juliet that likes to be offer to do some testing for you know, some new technologies being launched. So we're quite fortunate to be able to have that in-house and just keep scanning what's on the horizon.
That's right. Yeah, sort of looking out for new technologies that we can bring in, and then you know we're you know, really keen to sort of beta test new technologies. And
Yeah, great. And you know, you mentioned new technologies there, and you know that brings me beautifully into the next question, which is, you know, digital pathology and spatial biology. So I'd love to hear a little bit more about how you're using those techniques.
So yeah, so the institute, as Julia mentioned, when it first opened up, there was the in situ aspect of it, but there was also already a digital aspect of it. So they had a Zeiss Mirax and Leica XT, and also like the aerial fluorescent system. So they had those imaging platforms from the very beginning. They used ImageScope from Leica to do the analysis and things like that. So the Genie software, I mean, this is going back.
Long before my understanding of digital pathology, to be honest.
Yeah.
Sort of, so they were very early adopters of it, and then this has then just sort of snowballed. So as new things have come along, we've been quite early adopters of Halo. So I think we got Halo not long after it was released. So it's sort of shaped and moulded how it's been
that's
driven.
right. So all of those different technologies that we've been working with along the way. So, you know, initially we started with radioactive in situ hybridisation, which is just black and white, you know. But obviously now we've moved into kind of multiplex.
Yeah.
Like, you know, running 12-plex RNA-scope, for example.
Cara, you mentioned you're an early adopter of Halo here. So, you know, I'm curious, how important is image analysis to all of your workflow?
So yeah, so within not just our core, but all of the cores in the building produce images, quite a lot of different file formats. So you've got the IMCs and things like that. All these provide images, and it's great that you can actually get sort of quantitative and qualitative data out of these images for the researchers. And obviously all of our researchers have different levels of expertise in image analysis. So some of them are able to script and do their own complete things solo and are absolutely fine. Others are not computational based and therefore require the assistance of other sort of applications. So we have the Halo platform, we have VisioFarm, there's QPath, which is freeware available, there's a variety of different options, and we then provide a service. So we will either provide them a service of doing the actual analysis for them, or we can train them in the different technologies so then they can do it themselves. But it just helps them get the results that they need, and we can advise them accordingly.
Yeah, absolutely. I do want to move the conversation on to spatial biology, and perhaps, you know, image analysis within digital pathology is something we can come back to. But you know, we've mentioned it a couple of times, and you know, our audience are really very familiar with digital pathology, but perhaps not spatial biology. So, as you know, what I would consider experts in this field, you know, I'd love to hear a little more about it. Maybe you can introduce the topic.
Yeah, sure. So, I mean, spatial biology is that sort of not just like what's happening, but it's also the where is it happening,
Okay.
and like what cells, who with.
Sure. Okay.
So, you know, typically it's thought of as quite high-plex. Now it's multiplex. We're looking at, you know, lots of maybe protein markers and investigating those kind of cell-to-cell interactions. But not only kind of in a sort of, you know, not only proteins, but also in a multi-omic way. So kind of spatial biology is really a kind of an umbrella term for multiple different modalities. So we've got spatial transcriptomics, where we're, you know, looking at the gene expression within cells, but also then we've got spatial proteomics, so like your opal, like a comet, for example.
Yep.
And then maybe also other technologies such as codecs, not that we have them here, but and then finally there's kind of multimodal,
Okay.
so where we're looking at both RNA and protein together in the same section.
Yeah.
So, I mean, we've obviously been trying to keep at the forefront of those technologies here, and we have quite a few in-house.
Great.
Yeah.
And you know, talking about technologies then, so you've obviously, you know, got RNA scope, base scope, high-plex, DNA fish, an integrated RNA plus protein co-detection.
Yeah.
So, you know, which of these approaches, you know, proves the most useful in understanding the tumour microenvironment?
That's a really good question. I'm glad you've asked it, because actually it really depends on the question that the researchers are asking.
Okay, right.
So, you know, and as you know, we're supporting those researchers in lots of different projects. Actually, it's hard to pinpoint one particular thing that is the most kind of useful. I think it very much depends on the question that's being asked.
Yeah.
So what I would say is that we are finding that our requests are getting more complex and more specialised.
Okay.
We are tending to work a lot more towards you know, this kind of multimodal approach, where we're looking at RNAs and proteins on the same slide, and even like DNA and protein, which has been less common in the past. But yeah, I find that maybe a lot of the in situ hybridisation requests I'm getting now are, you know, they're a lot more specialised, a lot more focus.
From a cost perspective then, you know, is there a real jump from going from just, you know, digital pathology image analysis to spatial biology?
Yeah, I would say so. I think there's a big investment in digital pathology initially, but then because it streamlines processes, you kind of, it proves to be cost effective long term. Whereas for spatial biology, like, there is a big investment in terms of the equipment, but also the reagents, and it's turning out to be so expensive that, at least what's happening here at the building, is that users are being charged, or researchers are being charged per slide or per project, and the cost, like sometimes those platforms even integrate the digital pathology side of things, because it does the scanning, which kind of dilutes a little bit of the cost. But comparing one to another, I think, I'm not sure if you guys agree with that, but I think it's a massive chunk of their budget goes towards that.
We're finding a lot these days that you know, researchers that are applying for grants, they're including this stuff in their grant and coming to us for costing ahead, so that, you know, they can really make sure that they are accounting for the cost of spatial biology in these grant applications.
Yeah, great.
And the equipment, so it is a big investment, and obviously there's a variety of different options, so it's picking the best option for your institute and researchers. So it's, you know, because once you have it, that's where your investment is, that's where you've gone, you need to make sure you've got the right option, right tool for multiple jobs.
I guess you can start with the basics as well, and then see where your research needs to go in terms of lowering your plex at the start, and assessing whether those investigations make sense to carry on higher plexity and move on to the most expensive technologies.
Or lowering the resolution.
Or lowering the resolution.
So looking at sort of, say, using the Visium protocol rather than the Xenium protocol. The Visium is a slide. You have the, most people have the next-gen sequencers and things like that anyway. Yep. And then you're imaging it. You're just using a normal H&E for the scan. So that's a way of doing it without having like a really expensive piece of kit. And then you can see if that's actually going to give you sort of the results you want. And then you can start looking at, say, massive kit.
Yeah, okay, great. So, I mean, it sounds like you've got like a real variety of technologies, scanning modalities here.
Yeah.
I'm interested then, has that affected what research questions you can ask? Has it broadened your mind? You've got all of these different methods now of collecting information, understanding the spatial context on a completely different level. How has that affected what questions you can ask as a researcher?
I think that people are being a bit bolder, and we are certainly quite open-minded to trying, different techniques. For example, the multimodal stuff I'm talking about. Actually working up the protocols to detect RNA and protein on the same slide. It's not trivial. It's depending on the sample and how it's been fixed and how you want to detect that. So I think we're becoming sort of a lot more focused on that. What's the word? Like the experimentation really of how can we best
tweaking protocols.
Yeah, exactly. Yeah, the sort of the optimization of these protocols to get the data that are most useful for the research groups.
Sure.
I think the sort of big multimodal options are like sort of like throwing a net out there and then like seeing what you get, and then you can use that to then sort of go down in plex and then sort of investigate that in much deeper, sort of a deeper context.
Yeah, sort of such as Xenium, where you can investigate, like maybe 500 to 1,000 genes in one tissue section, and then perhaps validate those those results on a lower plex, with an RNA scope or a COMET panel, for example, or something like that.
Now, it's probably a good time to mention the fact that you guys have a bit of an event coming up here at the Cambridge Institute. I'd love to hear a little bit more about that. So, what's the purpose behind the event? Why are you running it? And what's going on?
Yeah, so it is the In Situ Hybridisation and Spatial Omics Symposium. It's on the 20th and 21st of May this year, so coming up quite soon.
Yeah.
QR code if you want to have a look. So this actually sort of came about from, in my sort of early career at CRUK. There were lots of kind of improving immunohistochemistry and, antibody validation conferences that you could go to, but I was really struggling to find anything kind of in situ hybridisation based. And my boss at the time, Will Howard, said, well, make your own then.
Sure, yeah.
And so I took on the challenge, and we hosted our first event in 2015. And the idea behind the symposium has always been that it's sort of technical focus, and really sort of covering a wide range of researchers. So, from early career up to, kind of group leaders.
Yeah.
And we really try to get some kind of high caliber speakers to kind of come in and share the approaches that their labs are using, but to enable sort of this, really kind of collaborative and networking focused event where people can, make new connections and really discuss the ins and outs of their protocols.
Yeah, absolutely. I think just to jump in there, I think that's just so important to this field right now.
Yeah.
These technologies are quite new, and a lot of labs are using them for the first time. So that collaboration and shared understanding of how to implement them and how to use them optimally is really, really important.
That's right. Yeah. So we're often sort of, we have panel discussions and we're talking about like the standards that we might be able to apply to in situ hybridisation. So obviously with the you know, the huge boom in spatial biology, the event has typically been always focused on in situ hybridisation, but we are moving now towards kind of a multi-omics kind of, you know, we're including more protein-based, you know, detection in this symposium. So yeah, it's across 2 days. We've got 4 sessions.
Fab.
And yeah.
Great. Thank you, Julia.
Yeah.